Okay, several of you asked me about the online assessment, so I've just put the dates up here taken from the Blackboard site. So as you can see, the first deadline is next Friday, and we've covered all the material that's relevant for that assessment, so you should go ahead and complete it before the deadline. And then the following deadline is 2 weeks after that, so that's material we'll cover in these last few lectures of mine. Also to say that the podcast on Tuesday was fine. I checked it myself and the recording and the visuals were okay. So today what we're going to look at is the secretory pathway, and in particular I'm going to focus on the structure and function of the Golgi complex, and then outline the basis, molecular basis for this process of vesicular transport. So how material is moved between the compartments of the secretory pathway. So if we come back and look at the process of how proteins that are synthesized in the cytosol end up in different compartments, then we can identify transport through the nuclear pore as one process, and we talked about that this time last week. We can identify translocation into and across the membrane, and we use the endoplasmic reticulum as the example that we looked at in most detail, although we also looked at the mitochondrion, and that's what we looked at on Tuesday. And then the third process is bivesicular transport. So that is where material that's been imported into the endoplasmic reticulum buds off in transport vesicles and fuses with other compartments of the endomembrane system or secretory pathway. And it's this process of vesicular transport that we'll talk about today. So, as we talked about, proteins can remain folded to at least quite some degree when they're imported into the nucleus because the nuclear pore is quite large. In contrast, when you're translocating a protein across a membrane such as the endoplasmic reticulum or mitochondrion, the protein needs to be in a loosely folded conformation. However, proteins that have inserted into or translocated across the ER membrane, once they're in there, can remain folded. So folded proteins can be transported inside these vesicles, and in fact that's typically what they do transport. All of these mechanisms require energy, and it'll be no surprise that that means either they consume ATP and or they consume GTP. So vesicular transport is also an energy requiring process that uses nucleotide triphosphate. So proteins and lipids that are made at the endoplasmic reticulum are typically then transported through this so-called secretory pathway from the ER through the Golgi complex to the plasma membrane or cell surface. And that's the process we'll look at, the exit from the ER. However, not all proteins are destined for the cell surface, and some of those proteins have to be retained at specific intracellular locations. So they enter the secretory pathway, but they don't make their way all the way along it. And before we look at the way that proteins get completely out of the cell, either into the plasma membrane or secreted, what I want to look at is how particular proteins are retained at a specific place within the secretory pathway. So what we're saying is there's information that stops them from completely leaving the cell or reaching the plasma membrane and keeps them at a specific point within that secretory pathway. And in essence, that relies on specific sorting or trafficking signals which are part of the protein. In this case, they don't confer delivery to the endoplasmic reticulum, but once the proteins enter the secretory pathway via the ER, they dictate whereabouts the protein will end up within the secretory pathway. So again, this comes down to, quite often, amino acid-based sequence information. So perhaps the best known example is the so-called KDEL signal. And this is just single letter amino acid code for lysine, aspartic acid, glutamic acid, leucine. And those 4 amino acids, when they're present at the C-terminus, or the end of the protein, the last part of the protein to be synthesized, lead to the retention of soluble proteins, and this is primarily chaperones, so folding factors that you want to keep in the ER. And the ones we've talked about are BIP, this is the ATP-dependent chaperone, and PDI that catalyzes disulfide bonds. Both of those are soluble proteins present in the ER lumen, and they're kept there because they've got a KDEL signal. Now, in other cases, you need to retain membrane proteins within the membrane-enclosed compartments of the secretory pathway and not allow them to be trafficked to the plasma membrane. And in some cases, this relies on the size of the transmembrane region. So a typical membrane protein in the plasma membrane will have an alpha helix that's made up of about 20 hydrophobic amino acids, and that will be the transmembrane anchor. If that transmembrane anchor is slightly shorter, and by slightly shorter we're talking about 17 or 18 amino acids instead of 20, then proteins such as that are typically retained in the Golgi complex and don't make it all the way to the plasma membrane. Then finally, I alluded to this already when we talked about N-glycosylation, and we'll come back to it again next week. The addition of a mannose-6-phosphate to N-linked glycoproteins synthesized at the ER can occur in the Golgi, and the addition of that mannose-6-phosphate leads to those proteins being diverted to the lysosome rather than being secreted from the cell. So if we go back and look at a schematic, what we can see is if newly synthesized proteins enter the secretory pathway, then should they have a KDEL signal, that will park them in the endoplasmic reticulum. If it's a membrane protein and it has a slightly shorter than normal transmembrane domain, they'll end up being resident in the Golgi complex. And if they happen to have a mannose-6-phosphate tag attached, they'll be diverted from this pathway to end up in the lysosome. So, if that's true, then what does that mean in practice? So, let's think about a hypothetical situation. If we took the DNA encoding for one of the ER-resident chaperones, such as BIP, and we removed the KDEL retention signal from the end of the protein by site-directed mutagenesis and expressed that protein in a cell, what do you think would happen to that protein? Where would it end up? Yeah, so prediction would be it will be secreted from the cell because it no longer has that information to keep the protein in the ER. So the converse of that would be we take something like insulin that's normally secreted and we now extend it slightly and add the KDEL sequence to the end of insulin. So what's now going to happen to that insulin molecule? It'll stay in the ER. Yeah. And these are exactly the experiments that were done to prove that the KDEL signal was an ER retention signal. So, when proteins leave the endoplasmic reticulum, their first port of call is in essence the Golgi complex. And the Golgi complex has been studied for a long time because it's very clear when you look sort of down a light microscope. You can see it as this sort of stack of cisternae, or plates, often located quite close to the nucleus. So these are referred to as the cisternae, or stacks of the Golgi. And it's important to know that the Golgi has two distinct faces. So it has what we call a cis, or entry face, where material comes in from the ER. Then we've got a bunch of specific and distinct cisternae. And then we've got the transface, or the exit face. So material is coming from the ER to the cis face of the Golgi, moving through the Golgi, and leaving the Golgi at the transface, or the trans-Golgi network. So, as I've said, newly made proteins will leave the endoplasmic reticulum in these little transport vesicles, and we'll come back and look at this in a minute. They'll enter the cis-Golgi network, or the CGN, and the cargo proteins will then move through the different cisternae of the Golgi and eventually make their way to the trans-Golgi network. They'll leave the trans-Golgi network in transport vesicles, and we'll look at that next Tuesday, and eventually be delivered to the cell surface. So in very crude sort of cartoon form, that's this process.
Here we see a Golgi apparatus, with its cis face toward us. We also see vesicles and several microtubules. A small vesicle, shown in red, is moving along a microtubule carried by a motor protein. Materials move through the several cisternae of the Golgi apparatus. They are carried in vesicles that bud from one cisterna and fuse with another, or with the plasma membrane.
So, material is transported from the ER to the cis-Golgi, through the Golgi, and leaves the Golgi at the trans-Golgi network. The model I just showed you is one of two models that are used to explain the way that material moves through the Golgi. So the model we looked at was the so-called vesicular transport model, where little vesicles bud off from one compartment and take material to the next compartment that they fuse with. There's a second model called the cisternal maturation model, where essentially the cisternae move with their cargo through the Golgi complex and mature. And if we compare those, essentially the difference is one of these involves the sort of essentially the equivalent of the movement of the cisternae in terms of position through the Golgi, and the other involves movement in the vesicular, bivesicular transport. One of the reasons that it was necessary to develop a model that was different to or additional to the vesicular transport model is that some of the proteins that are transported through the Golgi are just too big to fit into these little vesicles. So these little vesicles are about 100 nanometers in diameter, and some molecules like collagen are too large to fit. So current view is that probably both aspects of these models are correct, and some proteins will use a sort of vesicular transport route, and other proteins, for example, large proteins like collagen, will use a cisternal maturation type route. What's the point, right? So why put material through the Golgi? Why not just take it straight to the plasma membrane and make life simple? The Golgi has a number of specific functions. So material moving through the Golgi undergoes a variety of processes, and that's the reason why material is transported through the Golgi. So actually the first important function of the Golgi is protein sorting. So whilst we talked about the KDEL signal as a retention signal, it's a retention and retrieval signal. So sometimes a protein with a KDEL signal escapes from the ER, gets into a transport vesicle, and gets transported to the cis-Golgi network. In those cases, that protein is recognized because of its KDEL signal and brought back to the ER. So that process of protein sorting is distinguishing proteins that should be retained within the secretory pathway, for example, at the endoplasmic reticulum, and distinguishing them from cargo proteins like insulin that should be secreted. And that's one of the key functions of the Golgi. Secondly, the Golgi carries out protein modifications in the same way as the ER carries out protein modifications. So, as I already mentioned on Tuesday, these high mannose N-linked oligosaccharides that are added to proteins in the ER are altered in the Golgi. So they're trimmed back and built up with different sugars. In addition, the Golgi also carries out glycosylation itself. It's a different type of glycosylation. So in contrast to the N-linked glycosylation that happens in the endoplasmic reticulum, this is O-linked glycosylation. And this is the addition of oligosaccharides to some hydroxyl groups of proteins as they move through the Golgi. So O-linked glycosylation can be added to the hydroxyl side chains of serine and threonine residues so that these residues can become O-glycosylated, so the protein will be O-glycosylated. And I'm sure I don't have to tell you that serine, threonine, and tyrosine all have hydroxyl groups, but it's only serine and threonine that can be O-glycosylated, and tyrosine can't be O-glycosylated. Okay, so all three of these amino acids can be phosphorylated, but only serine and threonine can be O-glycosylated. The nature of the glycan and the glycosidic link is also different. So this is the N-linked linkage that we looked at last time, and this is the O-linked linkage that we're talking about now. So we've got a different linkage of a different sugar to a different amino acid side chain. So they're both forms of covalent modification with carbohydrates, but they're quite different in their sort of chemistry. So, exam question from a few years ago now. The known functions of the cis-Golgi include, hands up if you would vote for protein sorting. Okay, good. That's the right answer. Right. So, we've got these different compartments of the secretory pathway, and it's clear that material moves in these little membrane-enclosed vesicles between the compartments. So these so-called transport vesicles link the compartments of the endomembrane system and the plasma membrane. And today what we're really looking at is the outward or secretory pathway. So material on the way out. We'll look at material on the way in next week. So these transport vesicles will carry newly made proteins and lipids. So as I mentioned last time, the endoplasmic reticulum is also a major site of lipid biogenesis. And it'll take those cargoes from the endoplasmic reticulum through the Golgi, for example, to the cell surface. And the nature of these transport vesicles and the components that regulate them has been studied in great detail. And in 2013, Jim Rothman, Randy Schekman, and Thomas Sudhoff were awarded the Nobel Prize for their discoveries of the machinery regulating vesicular traffic. So essentially, the detailed information that underlies the brief thumbnail sketch I'll give you now. So this is very sort of topical work. So the basis of vesicular transport is the following. If you've got a donor compartment here, for example, the endoplasmic reticulum, and we've got a target compartment, let's say it's the cis-Golgi, material buds off from the donor compartment, forming a membrane-enclosed vesicle with cargo, and it fuses with the target compartment to deliver that cargo. So vesicles loaded with cargo from the inside of one compartment will pinch off and fuse with the membrane of the next compartment. This does two things. Firstly, it delivers the content, the inside, so the red dots in this case, i.e. proteins like insulin or other secretory hormones, enzymes and so forth. Secondly, as you can see, it also delivers the membrane. So the membrane's budded off here, and upon fusion, that membrane becomes part of the target compartment. And the membrane involves both integral membrane proteins and lipids. So it's important to realize that the content is delivered and the vesicle, and both of them become part of the target compartment after fusion has occurred. And those materials are delivered from the first to the second compartment. So if we think about the same thing in terms of secretion or exocytosis, which we'll look at again in detail next week, after fusion, in this case, the vesicles become part of the plasma membrane, and the content of the vesicle, let's say insulin, has been released to the outside of the cell. Another important feature of membrane fusion and membrane budding is that membrane's orientation is retained. And the simplest way of thinking about this is that whatever faces the cytosol during formation of the vesicle will continue to face the cytosol after fusion, either with another intracellular compartment or with the plasma membrane. So this is consistent with what I told you when we talked about the origins of the endoplasmic reticulum in the endomembrane system. The lumen, or the inside of the ER, is topologically equivalent to the outside of the cell. So anything that faces the lumen of the ER, when that material reaches the plasma membrane, that will face outside the cell. So when carbohydrate is added to this region of the protein in the ER lumen, eventually that carbohydrate faces outside the plasma membrane, right? So this face of the membrane is cytosolic, and after membrane fusion, it's still cytosolic. Now, it goes without saying, I hope, that these little vesicles don't just randomly butt off one membrane and fuse with another one. It's a highly coordinated event that's regulated by proteins. So what I want to do is look at the process of vesicle budding. That is, how does a bud begin to form, and how does it ensure that it contains material or cargo that's to be delivered? So, based on a number of studies, primarily initially electron microscopy, it was clear that these budding vesicles have a characteristic protein coat on the cytosolic face. And hence, they were termed coated vesicles, because you could see that coat in an electron micrograph. This was before people had any real idea what these coats were made of. We now know that the function of these coat proteins is twofold, and this work is really what led to these Nobel Prize-winning awards that I was talking about. So the first function of the coat proteins is to help to shape the membrane and persuade it to start to form a bud that will eventually form a spherical vesicle. In addition, the coat proteins have a key role in helping to recruit or capture the cargo that's to enter the vesicle and be taken on to the next compartment. Now, when the coat proteins are on the vesicle, it can't fuse with the target compartment. So at some point after budding, the coat proteins have to be removed in order for the vesicle to fuse with the target compartment. So what you can see in the next slide is an example of the kind of electron micrographs that I'm talking about, where we can see the coat on this side and the cargo on this side of a forming vesicle. And in this case, we're looking at a clathrin-coated vesicle. Clathrin is one example of a coat protein. So there are different examples of coat proteins, and they're responsible for different transport steps. So we can see that better here, where we can see the key sort of coat proteins that have been identified are clathrin, which is the one I mentioned, which in fact is involved in the endocytic route, or the uptake of material from the plasma membrane, COP1 coat and COP2 coats. So different coat proteins will select different cargoes and create the formation of transport vesicles that will mediate different steps within the secretory and endocytic pathways. And we'll come back and look at that next time. The fundamental principles of the way in which the vesicles are formed is the same for all of these coat proteins. So, for example, we talked about ER to Golgi transport. This involves a COP2 coat. And interestingly, clathrin forms a very ordered structure, a sort of basket weave around the forming vesicle. So it's strongly involved in driving the formation of that structure through the sort of three-dimensional organization of the clathrin coat. So what I want to do now is just look at the steps involved in vesicle budding, and then I'll run a little animation at the end that sort of brings these all together. So step one of the formation of a vesicle would be the assembly of the coat protein on the cytosolic face of the membrane, and a vesicle will start to form. And in addition to the coat proteins, shown in dark green, in light green we've got adaptins. And the adaptins act as adapters between the coat protein and these blue guys here, which are cargo receptors. The cargo receptors span the membrane, so they've got part of the molecule facing the cytosol and part of the molecule facing the extracellular space, or potentially the inside of the lumen of one of the organelles, and they directly bind to the cargo. So the coat protein doesn't bind the cargo directly. The coat protein binds to an adaptin. The adaptin binds the cargo receptor, and the cargo molecule binds to the cargo receptor. So we've got a sandwich with the coat protein on one side and the cargo on the other, and a couple of components in between. That means that where the coat protein is forming, the coat cargo molecules are being selectively recruited into the vesicle, so we're concentrating the cargo for transport to a different compartment. Now, at some point, once the vesicle has almost formed a sphere, it needs to pinch off from the donor compartment. And that requires a distinct molecule called dynamin. And dynamin assembles around the neck of that bud, and with the help of some other proteins, again uses energy, so GTP, and the hydrolysis of GTP to drive the pinching off of that forming bud from the donor compartment to give you a free vesicle that's now been released from the compartment. Then, as I mentioned, the coat has to be removed to give you a naked or uncoated transport vesicle that's now competent for fusion with the target membrane. So the coat's important for the formation of the vesicle and the recruitment of cargo, but after the vesicle has been released from the donor compartment, the coat has to come off so that the vesicle can then fuse. So let's just run this little overview of that process in relation to clathrin-coated vesicle formation.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles. This movie shows a series of electron micrographs that have been artificially morphed to show the process of endocytosis as it may occur. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions.
So, although that's talking about material being taken into the cell, essentially the process of vesicle formation within the cell is identical. The fundamental components are the same. Now, after the budding of the transport vesicle and the release of the coat, it's got to deliver its contents to the target compartment, and it's got to reach the correct target compartment. So the vesicle is typically transported along microtubules towards the target compartment. And the recognition of these transport vesicles by the correct target compartment relies on additional proteins. And there are many. And again, a lot of this work comes from these Nobel Prize-winning researchers that I mentioned. But amongst the key players in this process are firstly the Rab proteins. And these again are small GTP binding proteins. And secondly, the snare proteins, which are membrane proteins. And together, the Rab proteins and the snare proteins ensure that the budded vesicle fuses with the right compartment and doesn't fuse with the wrong membrane. So if we look at the Rab proteins, sort of identification of vesicles by the target compartment relies on unique combinations of Rab proteins and tethers. So vesicle identification will rely on the Rab proteins that's located on the vesicle surface, shown here in yellow, finding its partner tethering protein on the target compartment, and that interaction initiates the first step of the delivery of the vesicle to the target compartment. The next step of that process depends on the other components I mentioned, the snare proteins. So the snare proteins are integral membrane proteins, and they contribute to the specificity of the fusion event after that initial Rab-mediated recognition step. And we find two types of snares. So on the vesicle, we have so-called V snares, or vesicle snares. And on the target membrane, we have so-called T snares, or target snares. And those snares recognize each other and give you specificity of interaction. So the V snares interact with their complementary T snares, and they dock the vesicle in place. So that vesicle is now sort of tightly docked onto the target membrane and is primed for fusion, so primed to deliver the membrane and the content to the target organelle. The final step is fusing those two membranes together. Now, we already mentioned that when we want to release a budding vesicle, we have to consume energy. So it's probably no surprise that this process of fusion also has to be driven in some way. And that membrane fusion is energetically unfavorable. And one of the contributing factors to pushing the equilibrium in favor of fusion is that these snare proteins twist around each other and essentially pull the two membranes together. So the properties of the snare proteins and the formation of these tight interactions drives the equilibrium in favor of membrane fusion. In addition, the fusion of the bilayers requires additional components, not shown here, called the membrane fusion complex. So there are other factors, other proteins that facilitate the mixing of the bilayers such that now this vesicle has become part of the target organelle or target membrane. So to finish up, what I want to do is just run a movie where we follow a fluorescently tagged protein from the endoplasmic reticulum through the secretory pathway.
Fluorescently labeled membrane proteins start their journey to the plasma membrane after synthesis in the endoplasmic reticulum. They are first dispersed throughout the extensive membrane network of the endoplasmic reticulum, from where they move to exit sites that form in random locations in the membrane network. At each of these sites, the membrane proteins are concentrated and packaged into transport vesicles. Clusters of the transport vesicles fuse to form transport intermediates. At the next stage, transport intermediates move along microtubule tracks to the Golgi apparatus near the center of the cell. The membrane proteins exit the Golgi apparatus. They move in transport vesicles that are now pulled outward on microtubules, which deliver them to the plasma membrane. Each time a Golgi-derived vesicle fuses with the plasma membrane, its content proteins disperse.
So at that last step, when the vesicle fuses with the plasma membrane, the fluorescent label is essentially being diluted out as the protein diffuses out into the plasma membrane. So, if we look at a previous exam question from last year, role of adaptins is to bind snare proteins, all right, promote vesicle fusion during membrane transport, bind cargo receptors. Excellent. If you weren't here at the start, just to flag that the deadline for the first online assessment is next Friday. So, in summary, we talked about protein sorting in the secretory pathway, the structure and function of the Golgi, and the molecular details of vesicular transport. Next week, we'll pick up on secretion and endocytosis. If anyone has any questions, please do come and see me. Otherwise, I'll see you again on Tuesday.
Hi. So I got some questions.
Yes.
One is about basically